Our next speaker is Emily Rogalski, Northwestern University. The title of her talk is Neurobiologic Features of SuperAgers. Is it resistance, resilience, or just luck?
Okay, so I thought we'd try to have some fun here at the end of the day. Hopefully I won't regret putting these questions up here. Okay, so before we start, I'm really excited to be here. It's such an honor, and I'm excited to share with you about the SuperAgers that we study. But before I can kind of talk about what SuperAgers are, I thought I'd just give a brief amount of background. We all know this, but here's the depressing slide of the day where you look at age across the x-axis and then memory performance on the y-axis where higher numbers are better, and you see this over the decades that there's this decline. And so if we were to just look at these average data in isolation, we might come up with this conclusion that cognitive decline is an inevitable consequence of aging, and that we could merely determine someone's cognitive age by the passage of time. And by the many talks we heard today, we know that that's not adequate. And so one way to show that that's not adequate is just by putting the actual individual data up there, and then the story becomes much different. We see now the green area is the average range, and we see that that average range is very narrow back in our 30s and 40s, and it expands over time. But the thing that got us really intrigued were these people that I put in triangles here. So these individuals seem to be resistant in some way, and they're performing as well as individuals decades younger than themselves. And this was kind of the theoretical spark for the idea of studying what I'm going to describe as superagers. Okay, so now I can change that comment and say cognitive decline may be a common, but maybe it's not an inevitable consequence of aging. And so cognitive age is more likely a composite of genetic factors, experience, and the passage of time. So if we have these simplified trajectories where we have a pathologic trajectory, a normal aging trajectory where there's some loss of memory, and then perhaps theoretically there's this super aging trajectory. So that's kind of the framework we are under, but now we've got to operationalize it. And we did it in the following way. So we glued the word super and aging together, we put a capital A, and we wanted to distinguish it from successful aging. And we said, okay, these individuals specifically, there's an age requirement. They must be over the age of 80 because that puts them at greater risk by being a bit older. And then they need to have episodic memory performance that's at least as good as individuals in their 50s and 60s. For us, we use the Ray Auditory Verbal Learning Test. It's a wordless learning test. It's very challenging. You have to remember, you're given a list of 15 words, and then we see how many you remember after a delay. They need to remember at least 9. An average 50-year-old or an average 80-year-old needs to only remember 5 as comparison. Performance on other cognitive domains, we just require to be average for age. And so then what are we going to do? We're going to bring them in, and we're going to enroll them in a longitudinal project. We follow them every 18 to 24 months, and then we ask them to donate their brains at the time of death. So why should we study these individuals? We know that we're getting quite good at extending the lifespan, but that's come at a consequence of our health span not keeping pace. Superagers are a good representation of people who have a better balance between lifespan and health span, and that's what we're trying to achieve here. We also, this has also been recognized in the Alzheimer's community. So the National Alzheimer's Project Act, when there was a research summit in 2015, one of their major recommendations was to understand all aspects of healthy brain aging and cognitive resilience to inform strategies for Alzheimer's disease prevention. So one way to study Alzheimer's disease is to look at what's going wrong with the brain. The other way is to flip it on its head and look at people who are doing extremely well. That's what we're doing here. So how is a superaging study different from successful aging studies? So the idea of successful aging was so important, right? It got us thinking about what's going right with the brain rather than just all of the news headlines of all the things that go wrong, including our hair turning gray, our eyesight getting worse, our skin getting wrinkled. But the challenge with it is that in 2006, there was a review study done about different definitions for successful aging. And so I'm going to blow up the abstract here, and the authors identified 28 different studies and found 29 different definitions of successful aging. It's not that any of these definitions are wrong, but it makes it really challenging to start to compare across studies with all of these different definitions. And they also found, when they looked at the proportion of successful agers in these studies, that the proportion varied greatly, so from 0.4% to 95%, so essentially no one to essentially everyone. So our study is just a little bit different in that we didn't want to use the term successful aging to describe these superagers because we had these very specific criteria for age and memory performance. So we're interested in their cognition, and these are the things that we measure in this study. So we require them to have outstanding memory function, but we're also interested in other cognitive function and how they're doing over time. One way to look at neuroanatomy is with MR imaging, and we do that with both structural imaging, resting state, DTI, the usual suspects. Genetic factors, of course, are important in histopathology. We recognize that things are more important than these biologic cores, so we have data on lifestyle factors, psychosocial factors, family history, and education, and I'm going to try to share some of these initial data that we have so far. I say initial because we're still enrolling these individuals and following them. So the first question, we put together these ideas or this definition before we actually found the superager. So the first question was, could we find them? And of course we could, because otherwise I wouldn't have been invited. So just to convince you, on the orange bar, you can see a graph of, or the bar graph of the superager performance, and then next to that are the norms of 50 to 60-year-olds, and then the norms of 80-year-olds. And you see the superagers are indeed, we're able to find them, and they're doing at least as well as those 50 to 60-year-olds, if not a little bit better. You'll notice we've had a lot of discussion today about gender differences. There's more women than men in the superaging study. I'm not going to comment about women or men being better or more superagers. I do want to say two things about that difference. One is that could be due to the fact that women live a little bit longer, but I think we should also consider one other factor, and that's women tend to volunteer for studies more often. So that might be leading to some of the bias that we have in these data. Superagers are required to be age 80, but they can enroll any time after they're age 80. So we see that on average, the superagers are 83 years old, but someone enrolled just last year that was 101. The range of education goes from 12 to 20, so we're not just enrolling doctors and lawyers. So cognition. So you might have been a little bit suspicious. You could have said, well, maybe they had a good testing day, but can superagers actually maintain this performance over time? And what you see here, again, in the gray shaded region, is average to superior range to 50 to 60-year-olds. And we see that all the superagers over an 18-month period maintain their status of performing that well, except for one. And I'm going to argue that she had a bad testing day on that day, because when she came back 18 months later, and then another 18 months later, she soared right back up in that gray range. So we've got some good performance over time. And then there are also people who have been in this study for a bit longer. And these are data from a superager who we've been following for over 7.5 years. And you see that she continues to have this great performance over that 7.5-year period. So one of the first questions that we asked in the superaging study is, okay, we've got people who have outstanding memory performance. What do their brains look like? Do they look more like 50 to 65-year-olds who they match in memory to, or do they look more like 80-year-olds who they match in age to? And so we started first by making a comparison between average elderly and average middle-aged individuals. And we saw something that's been commonly reported throughout the literature. We see this widespread atrophy. So everything in red and yellow represents significant cortical thinning that's seen in the average elderly compared to the average middle-aged individuals. And we see this both on the medial surface and on the lateral surface. But what happens when we make that same comparison in the superagers? So the picture looks drastically different. This isn't a typo in my slides. There, in fact, was no significant difference between the superagers and those who were in their 50s and 60s. And then things got even more interesting when we looked at the lateral surface, and we found this region in blue, which is not something we expected. So this is a region actually where the superagers have thicker cortex than the 50 to 60-year-olds. And when I give this talk to the lay public, they often raise their hand and say, okay, what is that region and how do I get one? Should I eat more blueberries or drink more wine? And I'm going to tell you the same thing I tell them, is I don't have the answer to all of those questions, but I can tell you that region is the anterior cingulate, and we know it's important for a lot of different functions, including attention, and that it supports memory. And this fortuitous finding has led some of our subsequent work that we've done under the microscope that I'll get to in a couple minutes. But before I go there, the other question that commonly comes up is, well, is it that superagers, how do they end up with this thicker cortex? So if this is a cartoon drawing of a superager, of a normal ager with decline over time, were superagers born with bigger brains? And I'm going to call that luck. Or do they have some sort of resistance to age-related atrophy? And this is work that was done by Amanda Cook, who's a graduate student in the lab, and we're looking at cortical volume over 18 months. So we don't have the luxury of going back in time and having MRI scans of each of the superagers from their 10s, 20s, 30s, and 40s, but we can look at atrophy over time to see if they're on a different trajectory. And what we see here is just that the rate of volume loss in the average 80-year-olds is over twice that of the superagers. So there seem to be on sort of a different trajectory of decline as far as cortical brain volume goes. So then if we go on now to histopathology, we've done a lot of different work, and I only have time to highlight one of the things that we've done. And so the question we asked here was, with respect to the anterior cingulate, were there more von Economo neurons? And this may seem a little bit random, but we started actually paying close attention to the anterior cingulate after we had the MR finding. And so I'll just briefly walk you through why von Economo neurons. So they're a very large cell, and they're primarily been described in the anterior cingulate and frontal enteral cortex. And they've also been only described in higher order species. The loss or abnormal development of von Economo neurons has been described in autism, schizophrenia, bipolar disorder, and frontotemporal dementia. And this is work that's led by Cengiz Giola, who is a co-PI on the SuperAging project. And so when we were just looking at the anterior cingulate, we noticed there seemed to be an abundance of von Economo neurons. And so then we enlisted a graduate student, Tamar Geffen, to go in and count the density of these neurons compared to average agers, and we found that superagers showed more than 4 times the number of von Economo neurons relative to elderly controls. Genetic factors. So we've started doing whole exome sequencing in superagers, and I don't have any data to share with you today. Another thing we've talked a lot about today is ApoE. So we see in the superagers that there's a slightly lower percentage of individuals who have at least one E4 allele compared to an average cohort of average agers. About 20% we see have at least one E4 allele. And so these might be an interesting cohort when we start talking about this idea of escapees. There doesn't seem to be an overrepresentation of E2 in this group, and there's no one who's E2. Psychosocial factors. So we have each of the superagers fill out the NEO, a personality inventory. So the NEO stands for neuroticism, extroversion, openness, agreeableness, and conscientiousness. And we saw some NEO data earlier today. In our superagers, we see that they have lower neuroticism, higher extroversion, and higher agreeableness. So in summary, we asked the question, is it possible to have individuals who have preservation of mental sharpness over age 80? And we say, yes, superaging is a rare possibility. And I put the word rare in there because it took us a long time to find these individuals. We've screened over 1,000 people who call us and say, hey, I think I'm a superager, and we find that far less than 5% end up qualifying for the study. That's not to say they're not superagers, and how we tell them that is a whole different discussion. And so then, are there some biological factors that contribute to being a superager? And so yes, there appear to be some biological factors, and I shared some of those with you today. And so if we go back through and play the game, is it resilience, resistance, or luck? So better cortical integrity, we see that longitudinal data sort of leans toward the idea of resistance, although we can't rule out the fact that they had bigger brains when they were younger. More Venns, I'm gonna go to the camp of luck. We have some data that suggests that superagers actually have more von Economo neurons than a younger cohort. So we're gonna say that that's more, they were born with it. ApoE, maybe we've got both some resilience and luck going on. So for those who have an ApoE2, maybe that's the luck. And if we go with the individuals who have an E4, maybe they're somehow resilient. And then personality profiles. I put resilience up there, and that might not make sense until I tell you a story. And that is that many of our superagers report that they didn't necessarily have easy lives. So we have individuals who were in the Holocaust, individuals who lost children at a young age, had very stressful events in their lives, but yet they seem to approach life in a quite different way than you might imagine if you had something so catastrophic happen to you. And so maybe they have these unique personality profiles that might be resilient despite their life experiences. There's a huge team of people who helped make this happen, and I have a special thanks to them, of course, to our funding agencies and the SuperAgers. A few years ago, we got them together. They demanded a cocktail hour. They wanted to meet each other, and now they're demanding another one, so we're going to try that again this May. It was a lot of fun last time, and hopefully proves to be the same again this time. And I'm running out of time, but I think I am going to end by putting up some lifestyle factor data as people ask questions, just so you can see.
Okay. You're fine.
I'm fine? Okay, yeah. So there's a kind of a mixed bag when it comes to exercise. We see that some of them exercise a lot. Some are participating or leading their stretching or exercising classes. This is not a stock photo. The person in the front is one of our SuperAgers, and that's her leading her stretching class. And people who weightlift, and then others who will very firmly tell me, Dr. Rogalski, I do not exercise, I do not plan on exercise, and I only run when chased. Tobacco use is also was rather high. We were a little bit surprised by that. Drinking is another mixed bag. So we sometimes ask people, why do you think you are a SuperAger? And we get a response occasionally that says, well, it's because I drink a martini every day at 5 o'clock. So I think that would encourage us to have a drink at the cocktail hour, maybe. Many of the SuperAgers, some of them are still working, and those who aren't tend to be very active in their communities. So they're volunteering, and we even have people who have gone back and gotten a second career because they were bored. And then blueberries.
Yes or no? Okay.
I don't know.
Thank you very much for a great talk.
So that was super impressive, and your cohort's super impressive. The people are super impressive. You alluded to this a little bit with your brain imaging data. I'm wondering, with your cognitive data, you're defining the group by comparing them to normative data in people in their 50s, right?
Yeah.
And you don't have data from when they were in their 50s.
Correct.
But have you considered comparing the discrepancy between their cognitive scores and some measure of premorbid is the wrong word, but earlier life IQ or things like that compared to other people, and to see if the discrepancy is different?
So we do give them all a WACE, and so we do have an estimate of IQ, and for each of the control groups that I showed you today, everybody's matched. So we can't attribute the differences that we saw between the average agers and SuperAgers, or average 50 and 60-year-olds and SuperAgers, to IQ, because those groups are always matched.
Right, but is the discrepancy between their cognitive performance and their estimated IQ different in the SuperAgers compared with the not, suggesting that there's really been less decline in the SuperAgers, and it's not just declining from some super high premorbid ability?
I'll have to look.
Do you know what I mean?
Yeah.
Okay,
yeah.
thanks.
Hi there. You considered, and Adam touched on one of the reserve proxies, but we know there's at least 4 reserve proxies, and you touched on 2 of those. I was thinking about premorbid IQ, but also leisure activities, and specifically leisure activities because we know with the experience score, they've done work where they've shown change in ACC activation over a 6-month period with cognitive activities. So just wondering.
Yeah, we haven't looked at the difference between leisure activities between groups, or
Other questions?
Do you worry that if you pick people at a certain sort of, and it is by definition, it has to be kind of an arbitrary distinction that you make, that you're losing information about the kind of the broad association that might exist in reality? Or let me ask it another way. Do you think that SuperAgers are a kind of qualitatively different category that carves nature at its joints, or do you think that they're the kind of high end of a kind of latent distribution of aging?
Well, the idea was that if we pick people who are sort of at that very, very tail end of the bell curve, or are at that elite end of the spectrum, that they may be able to offer us some different clues about which directions to look in next. It certainly is not a substitute for, it's not that we shouldn't look at lifespan studies, it's not that we shouldn't have cohort studies or population studies, but sometimes targeting a really specialized group could offer some new directions for understanding cognitive aging in those who are aging extremely well.
It's a very super interesting and great work. Congratulations.
Thank
So
you.
I have two quick questions. First, you mentioned in this group of people, the E4 carriers is slightly lower. Is there any E44 homozygous present in this cohort?
There's not.
Okay. Secondly, do you know about their brain A-beta deposition or tau-related imaging studies on this?
Yeah, so we're just starting to get some amyloid PET data, and we don't have enough to talk about any group data, but there are some people who have elevated amyloid.
Thank you.
Okay, thank you. Last two, go ahead.
So it might be informative to look at the children of the superagers compared to children of 80-plus-year-olds who aren't superagers.
Yeah.
And that might give you some clue about what they might have been like earlier on.
Yeah, it's a great
The other thing is,
suggestion.
do you have any guess as to if the superagers are likely to? My hunch is they're going to live a lot longer than the other 80 or 80-somethings, so that part of this has to do with longevity.
Yeah, we'll see. I mean, it's interesting, both the normal agers and superagers that we follow in this study, I think maybe an incentive to enroll is that they don't die. So that's good news for me because I run the cognitive ends of the study, and bad news for Dr. Giola, who is waiting for their brains. So far we haven't seen that, but it's early.
Okay, thank you. Last question?
Yes. What is your dropout rate?
We've had very few people drop off, and we've had people move, and so we follow them then with like phone interviews, and I can think of one other person who's dropped out.
The reason I asked that question, you have such a scintillating personality. I wonder how many of these people are trying to please you, or trying to remain in the program. I think there's some sort of relationship that is involved in this study.
Thank you very much. Great comment. Thank you, Emily.